Okay, so hopefully she'll be able to jump in at some point. Thank you for pointing that out to me. Where are we at? So, how do you wish to be tested on your vocabulary? Any thoughts about that? We can do some sort of matching. We can do some sort of fill in the blank. You can draw pictures. You can describe right by definition. What are your thoughts on all that? One vote for matching. Any other thoughts? Any other preferences? Would you agree with Hannah then? Yeah, okay. Okay, any other hard copies for turning in the assignment? I don't have assignment 2 up on Blackboard yet, but we'll take care of it. Hopefully right after class today, so. Okay, thinking about vocabulary pairs as a review. We got superior and what does that pair with? Inferior. What does caudal pair with? Cranial. Anterior with posterior. Ventral with dorsal. Lateral with medial. Proximal with distal. Superficial with deep. Flexion with extension. Abduction with adduction. So we went through compounds, we went through water. We should be starting with carbohydrates now. So we got some overlap with your things there that may or may not work. So where does yours, that new set start with? I got a weird overlap here. So we've gone through, so it should have some carbohydrate stuff there for you to get started on. So carbohydrates. So the name comes from, we're dealing with carbon, that C up there, and then you have H2O. The ratio of 1 to 2 to 1 is what you're going to find characteristic of carbohydrates. So you've got, so they came up with the name carbons, basically hydrated or hydrated. So if we're thinking about what's in this, we're adding oxygen from hydrocarbons. So we're going from carbon and hydrogen with the hydrocarbons over to carbon, hydrogen plus oxygen. So this should be, I think, is that in there or not? Yeah, so we're a little behind because I didn't get it to you. But if you want to take a moment, fill all that out. So we'll go to, I think there might be 1 or 2 of those there. So we're getting to carbohydrates. So generic chemical formula, as I mentioned already, the ratio of 1. So the N represents 1. We've got 2N for the hydrogens and 1N for the oxygen. So 1 to 2 to 1. So carbohydrates are organic compounds. There's quite a range involved with them. We go from sugars, which are monomers. These are the things that we'll link together that we talked about last time. Most of the time in nature, you're talking about linking glucose. So the sugars are also called historically monosaccharides. So you're going to have those there. The number of carbons in the hydrocarbon backbone can range from 3 to 7 in the body. The vast majority of what you're going to find have 6 carbons or 5 carbons. You'll have the other ones here and there, but the vast majority will be 5 carbon chain through the backbone, or more likely a 6 carbon chain through the backbone. So 6 carbons are the most common, then the 5 carbons, and then the other options, the 4, the 3, the 7. It's a huge polysaccharide. So we take those monomers and we put thousands and hundreds of thousands of them together to form starch, to form cellulose, to form glycogen in the body. We store those carbohydrates. We use those carbohydrates for various functions. So the string of monosaccharides are called polysaccharides. So we take those chains of monosaccharides, we call them polysaccharides. So on the planet, or at least on this planet, most of that is going to be glucose. So that is probably the most abundant organic molecule on the planet. You're going to have it as the base of life. Photosynthesis generates glucose, and that moves everywhere through the environment kind of as a currency among living organisms. Those polysaccharides can be branching. Some forms of cellular or some forms of starch will branch in a number of different ways. Cellulose tends to focus on chains. We're laying down chains of polysaccharides, sticking them together, and they wrap around the cell to form that cell wall. You're going to have lots of variations in between. So when we're thinking about sugars, we're going to use the suffix -ose. So 5 carbon sugars, pentose, 5 sugars, 5 carbons in that sugar. Hexose for 6, so 6 carbons in that -ose designation. Others exist. There's a whole lot of different variations on the thing. There are many isomers in each of the 6 carbon groups, the 5 carbon groups, and as we go through. So as we get more and more atoms in a molecule, how we rearrange them, how we might put them together can be more and more complicated. So if we look at some of the isomers of our 6 carbon hexoses, these all have the same number of hydrogens, the same number of carbons, the same number of oxygens, but they're rearranged in a number of different ways. So glucose up here, those of you who haven't had chemistry, the intersection here, there's a carbon sitting there. So you've got 1, 2, 3, 4, 5, 6. Galactose, does anybody remember from Dairy Bowl or Dairy Jeopardy what galactose does? You take a galactose, you put it with a glucose, you get lactose. You take those two, stick them together, you get milk sugar. Fructose plus glucose is table sugar, sucrose. So disaccharides, which we'll get to in a second. So all have that 6 carbon, 12 hydrogens, and 6 oxygens for a chemical formula of C6H12O6, but they're rearranged in a number of different ways. The difference between, say, glucose and fructose is the sweetness. It interacts with the tongue slightly differently, given where the arrangement of carbons are. and you're going to get a sweetness associated with that fructose. So as I mentioned already, there are lots of different ways to draw these molecules or representations of these molecules. Anytime we're looking at a diagram here, the organic chemists get lazy, that anytime you see an intersection, there's an assumption that there's a carbon there. So you have that carbon backbone for glucose, 1 through 6. It can fold in on itself, the one reacting with the one carbon reacting with the 5th carbon. The OH, we lose water, comes out of that, and we have a circle now that's got 5 carbons in it and 1 oxygen. So the glucose will go back and forth between those two figurations, the circle and the straight line. So the ring or the chain, they are equivalent in the body. So why is glucose important? So it's the currency of life. So the first reason we have glucose is the main fuel in cells that they use to do work. So it's the gas that makes the motor run. It's the energy that makes the body run. Our dairy cows up at the dairy are going to be producing close to 4.5 pounds of glucose in their liver every day just to export as lactose. And we'll get to that in April. Second reason that glucose is important. Glucose and many other monosaccharides can furnish or donate their carbon skeleton that cells can use to manufacture other molecules. They become the base on which other molecules are built. For example, glucose can be used to make amino acids, which are the monomer for protein. The liver does a lot of that manufacture. Takes glucose, cuts it apart, take pieces of it. We can make amino acids with that. Third reason we might say that glucose is important is it can be stored for future use. So in nutrition, we talked about build or burn. Reasons 1 and 2, or 2 and 1, I guess the way I presented them. But we're adding a third use as we store it for later use. In our animals, we put strings of glucose together, and that is stored as glycogen for future use. Very similar to starch in its structure, but glycogen is in animals, starch is in plants. That glycogen will be found in the liver, it'll be found in the muscles, maybe a little bit in the kidneys, but the food coming in, if there's a whole lot of glucose, the liver will say, hey, I'm going to store that for later, trying to manage those blood glucose levels, moving through to get done what needs to get done. So very versatile. We can use it for energy, burn for calories. Structure, use it to build things or store it for later use. And it can go to either structure or energy later. So putting it aside, saving it for the future for those 2 original things related to glucose. So we're thinking burn, build, or store. So management of glucose is pretty important in most of our mammals. Being able to create that to keep the blood sugar where it needs to be, having that blood sugar to fuel the nervous system. The preferred fuel of the nervous system is the glucose. When we have to run on ketones and things, we usually get a little punchy, a little sluggish. And that's one of the problems with ketosis. We run low on glucose and we're not running the nervous system as well as we could. We take those monosaccharides, usually glucose or an isomer of it, and we can make disaccharides. So you've got, as I mentioned already, glucose with fructose. Those are isomers, 6 carbon units. Put them together, we get table sugar or sucrose, galactose and glucose. We put those together to get milk sugar. That's what the udder does every day. It takes glucoses from the blood, it isomerizes the galactose, changes it around, rearranges it, sticks it with the glucose, exports that lactose to make milk. And that's the solute that drives milk volume. Roughly, you could say, well, Holsteins produce more lactose than Jerseys for milk volume. So, maltose, when we break glycogen apart, we might get a disaccharide. That is 2 glucoses together. That has to be broken apart before it can be used in the body. So we're looking at chains that are anywhere from 2 sugars, the disaccharides, to thousands of sugars long, depending on what the need is, depending on where we're going. So if we're looking at glucose and focusing on that for this class, the polymers that can be made from glucose are starch, glycogen, and cellulose. You can see that on that diagram there. It's got the 3 different ones there. Starch is only in plants. Cellulose is only in plants. Glycogen is in animals. So we may be consuming, as our cows may consume starch and cellulose. Starch is very readily available, a storage form of glucose. Cellulose is a structural form of glucose that's a little tougher to deal with. So we need a whole system of digestion to make things happen. Glycogen is the storage form in our animals. So if we look at this diagram in your handout, we're not going to worry too much about what the names of things are and what everything's there. But you see starch, glycogen, and cellulose. What do you see as differences for those molecules, just looking at them up there as drawings? So what do you see as differences in that drawing?
Cellulose is always in a straight line.
So cellulose tends to form straight lines. The way they're linked together, you're going to have strips, chains that are very, very linear. What do you see with starch and glycogen? They tend to spiral around on themselves. They form coils. This is one of the reasons that there's a whole lot of water associated with starch. Think about potatoes. Because of that spiral pattern, water can fill up the center and be held in there. So the shape of the linkages, if you look at them, starch and glycogen, you got the same sort of pattern. They're very, very similar. Whereas something you're alternating here, and that alternation is what makes cellulose impossible for mammals to digest. They can handle the configurations on the top. They can't handle the configuration on the bottom, so they need help from the microbes to break those things apart. Glycogen and starch can form branches. You can see that in glycogen. So if you're tearing that apart, you need it to come apart quickly, which happens a lot with glycogen. So has anybody here played sports and your coach tells you the night before you gotta carb up? Pasta, spaghetti the night before? So you're trying to fill up your muscles with glycogen. If it's got a whole lot of ends, the branch points, you don't have to cut off one at a time. You can cut all the branches at the same time and release glucose to be useful, take it out of storage and make it useful very, very quickly with that branching. The enzymes can rip it apart a whole lot faster than it was just a straight chain. So we know that starch and cellulose are for plants, glycogen is for animals. We know the top two are storage. We can take it out for later and build and burn with it. Once cellulose is committed in a plant, it's not coming back. That glucose is committed to that function. The plant can't undo it either. So writing that down, polymers are made of glucose. Starch is storage in plants. Glycogen is storage in animals. Cellulose is used to build irreversibly in plants. They can't undo that after the fact. So as we move forward, remember glucose, remember glycogen. We'll be coming back to those when we talk about metabolism in the body and how do we get from feed in a cow to milk. So you should know by the end of the semester how milk is made inside the cow. So if you want to pull out your letters for a second, make sure we've got our tonicity down or osmolality. If an animal cell, for example, a red blood cell, is placed in a hypertonic solution, what's going to happen? So thinking about the answer, what's going to happen if you put that cell in a hypertonic solution? Anybody remember? If you can answer that, then the other ones become a little easier. The answer, picking the answer. What do we know about a hypertonic solution versus the inside? That solution is higher in what? Salt or solute, right? So that's the most common stuff we see, the electrolytes. And then water concentration outside in that solution is higher or lower than the inside. The hypertonic solution, if it's got more solute, higher, then water has to be lower. If you think about the hypertonic solution, the cell goes in there, it shrinks. If the solute doesn't move because you got the semi-permeable membrane, water is going to go from a high to a low. So you've got water concentration is higher in the cell than outside the cell. It will leave the cell shrinking that. So which of those up there fits that? D. Is this starting to become a little bit more easy to think of and do? Getting there, right? You still have to think a lot about it. So the opposite, the hypotonic or hypoosmotic, what's going to happen? Going to be the opposite of what we were before. So that becomes B, right? Lower solute concentration by definition. Therefore, it's got to have a higher water concentration outside the cell. So what's happening in the illustrated of exploding and shrinking cells? What's moving and what's not moving? Does solute move? No, because of the semi-permeable membrane. Does water move? Yes. So which one's that? C is the answer we're looking for. So remembering what direction the water goes, net flow, hypotonic, higher outside, lower inside, net movement is in. Hypertonic, water concentration is higher inside the cell, net movement is out of the cell. So thinking about how we learn, we could think that learning is like this, where a learned scholar just pours information into your head. Pretty boring, isn't it? Thank you for your honesty. What you need to find or get to this semester is more of an active learning, where you as students are interacting and helping each other learn. So information comes out for me when you're working on your assignments. Maybe you start to work together and look up when you're studying or in the review sessions. An interaction between the students will create a learning environment that's more robust and tends to stick longer. You don't forget everything right after the test if you can do that. So the 5 questions help with that interaction. So study sheets, if you guys fill those out together, that's an opportunity to work together. You've got that sample I gave you. You've got the Word file up on Blackboard if you want to print those out. So thinking about on your own for part of the time and perhaps working in a group, finding a way to make that work. So just some thoughts. So we've done water, carbohydrates. We're going to move on to proteins. So proteins are polymers of amino acids. We take amino acids, we link them together by what reaction? Dehydration reaction. We take a hydrogen from one side, a hydroxide from the other. We bring those together, pull the water out, and we form a bond between our 2 monomers. So if we're looking back through our diagram, so you might want to slide this one out of sequence. You got 2 of them now. We're adding one more of the big 4 as we go from carbohydrates to proteins. So we've got oxygen, hydrogen, carbon, but the key thing we're adding now is nitrogen. Nitrogen presence is one of the key definitions, key indicators related to our proteins. The fact that nitrogen is there makes it a protein. Everybody find their chart? So we're increasing in complexity as we work our way down. Water, pretty simple. Hydrocarbons are a whole lot bigger than each individual water molecule. Carbohydrates, we bring in that oxygen to make things a little bit more complicated. And now we're making things one step more, bringing in our functional group the amino group, which contains nitrogen. So the addition of the amino group gives its proteins its unique character. Those of you that had nutrition, we do crude protein as nitrogen times 6.25. So if we have the percent nitrogen in the feed, we multiply it by 6.25. That gives us the percent crude protein, crude in the sense that it's roughly right for a feed. So proteins, as we mentioned, are polymers of amino acids. The amino acids are the monomers. And the amino acids, I'm going to abbreviate as AA or AA small or lowercase s. So that just seems to be the way nutritionists do it. So if you see an AA, we don't mean Alcoholics Anonymous in this class, we mean amino acids. So proteins are made of amino acids. There are about 20 more or less running around that are used to make proteins. In our dairy cows, we divide them between essential and non-essential. So the essential, the body has to be provided through the diet. Non-essential, the liver can make. They all follow the same blueprint. We have an amino end, which centers around a nitrogen with 3 bonds. We have a middle carbon with what's called an R group attached to it. And then we have a carboxyl end at the other. So we have an amino group at one end, an acid group at the other. That's the same for all amino acids, that nitrogen, carbon, carbon. What's different among the 20 amino acids is the R group that hangs off that middle carbon. So it would look something like this. You have a nitrogen. We're not doing all the different things that are hanging off that nitrogen. There should be 2 OH groups. You're going to have another hydrogen hanging off that middle carbon. You're going to have the carboxyl group associated with that carbon. But we're going to focus on the backbone, what runs through a protein. Nitrogen, carbon, carbon. And then we have the R group hanging off of that middle carbon. That R group, whatever that is, varies from amino acid to amino acid. So we can have, for that R group, we can have just a simple hydrogen, which is glycine, a non-essential amino acid, to some very complex, huge hydrocarbon rings with many different attachments on them. So if you're looking at amino acids, the common part of every amino acid is that nitrogen, carbon, carbon backbone. What's different is that R group. And we have 20 different ones in the diagram we're going to look at here. When we make the backbone of protein, we string those amino acids together. The connection through, we have that common backbone where every amino acid connection, that dehydration reaction, is a red dash. So we've got amino acid after amino acid after amino acid. We're going nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. Every protein has that repeated pattern as its backbone. The question then happens is, once we've established that, how does the molecule rotate with the R groups, change shape with the R groups to form the final character of that amino acid? So this is oxytocin. Everybody heard of oxytocin before? Oxytocin is 10 amino acids long. So we start at one end of oxytocin with glycine. We follow the NCC, 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 common backbone that works all the way through. What's different is what hangs off that chain. You can see up top with the TYR, that stands for tyrosine. We got a big ring of carbons and an OH group hanging off there. So that backbone works its way through. What's hanging off is what gives the protein its character at the end of it all. So we've got that generic amino acid. We're putting in all the hydrogens and oxygens that are associated with that. To put two together, we're going to take a carboxyl end and we're going to dehydrate that with an amino end. So that dehydration reaction forms a chemical bond in protein. That bond is called a peptide bond. So between the carbon and the nitrogen, so the carbon from one amino acid to the nitrogen in the next one, that carbon nitrogen helps make sure we don't get things back and forth. Everything's lined up the way it's supposed to. NCC, NCC. We have a dipeptide, two amino acids together, di for two, versus a polypeptide or a short peptide going through. So that dehydration reaction removes that water to give us the bond. Again, that common backbone. This is a diagram. I think you have this somewhere with all of the different R groups. So we're just showing the generic amino acid up top. Then we have the R group hanging off it. So we've got 20 different, if you get the glycine up here. So if you look at the color scheme up there, a circle that is a white one is a hydrogen. We've got nitrogen. No, carbon is black. Oxygen is red. Yellow is sulfur. Blue as nitrogen. So we have three different classes up there. We have the hydrophobic. We have the blue section down there, hydrophilic. And then we've got the amphiphilic at the bottom. So amphiphilic as amphibian. What do we know about amphibians? They like water, but they can also get around on land if it's not too dry. So they're in and out of water. So they're somewhere in between. Hydrophobic at the top means it doesn't like water. Hydrophilic in the middle means it likes water. And then we've got some common characteristics going through. So looking at the hydrophobic run there, we've got alanine, valine, phenylalanine, proline, leucine, and isoleucine. What do you notice just looking about those ones that don't like water? No oxygen, no sulfur, tends to be mostly carbons and hydrogens. What do we call something that's made up of just carbons and hydrogens? Hydrocarbon. What do we know about hydrocarbons? They don't like water. We think about gasoline, we think about oil. Any of those don't like there. So we don't have oxygens in there that are going to lead to charges. Whereas if we look at the hydrophilic group, we've got nitrogens in there. We've got a lot more color, a lot more things that are going to lead to charges. The charges will lead to those R groups liking water. So if we look at hydrophobic, they're going to try to hide from water. They're going to try to group together and say, hey, I don't want to be anywhere near water. Kind of like oil, salad dressing, the blobs of oil grouped together. Those R groups are going to try to do the same thing. Whereas the hydrophilic are going to want to move with their charges toward water. And then you've got some things here that you've got some charged, not fully charged. They're going to do some different things somewhere in between. So basically, based on what it is, hydrophobic or hydrophilic is going to tell you how it interacts with water. So we're looking at proteins. We can think about them as long, thin structures or big blobs that are going to be like a big blob of spaghetti, or nicely laid out thin structural proteins like our tendons and our muscles. Those tend to be very stretched out. Proteins that contain the smallest amino acid, which is glycine, where the R group is just a single hydrogen, they can twist more readily, they can spin more readily along that backbone than the ones that have the big circles like phenylalanine. You can imagine that attached to something is going to make a whole lot of bumping and sticking as it tries to spin around, versus the glycine up top there. One hydrogen is not going to get in the way of anything. It can spin and turn all it wants to. But the bigger things are, the harder it is for that backbone to twist and spin. So more readily than consistently large amino acids in the sequence. So if you need some flexibility, if you're trying to make sheets, glycine becomes very, very important to lay things out straight and even. So we don't have the R groups in this picture, but we know that we can spin around that backbone. And our R groups, depending on the size, whereas where we have small group, R groups are probably going to spin with less interference than some of the bigger R groups, like phenylalanine or leucine. What happens is, depending on whether they're hydrophobic or hydrophilic, they're going to spin in opposite directions. They're going to want to twist toward water or away from water. So if we go through and we say that, sure I got the colors right, that the pink sort of matches our diagram with the hydrophobic, and then that blue section in the middle is going to be our hydrophilic. So we've got these R groups. We've got hydrophobic and hydrophilic. So what are the hydrophobics going to want to do with regard to water? They want to get away. The hydrophilics are going to move toward water. So the first thing that happens is they tend to get on opposite sides depending on what their preference is for water. So you have this protein forming, it's coming out of the ribosome. There, you're going to have the solution in the cell is water. You're going to have these hydrophobics, the ones on the top, are going to try to get away from water. And what they tend to do is huddle together. We have all the hydrophobic ones coming together, trying to create a little cell where there's no water in it. So in this case, they're going to take that backbone, that line in there represents what? NCC, right? So it's going to bend that up, twist everything around, and try to huddle together so they're by themselves. Whereas the hydrophilic, the red or the blue with the red letter there, they're going to turn toward water. They're going to be more happy next to water. So this reaction of the R group, whether it likes water, whether it doesn't, all those interactions will determine the final shape of the protein in that water solution inside the cell. If you put it in an oil solution, it would be a different shape. So if we think about our, has anybody ever heard of Pandora jewelry or not? You got one, two. You get a basic backbone, the bracelet, and you can add all kinds of beads on there that do or mean different things to different people. So each of these beads would be an R group and their interaction therein. So why don't we stop there? We'll take your break. We'll start again at noon and finish up. There's some stuff in the beginning of class, there's some overlap there, just throw out what you don't want.
Yeah, but that's stable.
Yeah, this one's smaller. There's only so much I can do with staples. Well, there's some redundancy there toward the end. You can decide which version you like, however you want to do it. Okay, going again. So once these R groups react with water, they're going to form that final shape. Is it going to be a sheet protein? Is it going to be a globular protein? Is it going to be some combination thereof? So when we think about protein structure, we think about different levels. The protein structure, the first level is the sequence of amino acids. So whatever string or backbone we have will ultimately determine everything else. So you've got that primary structure, you've got a sequence of R groups, that's going to determine how that folds. So primary structure is called, or related to the amino acid sequence. We have a diagram here that shows you all the abbreviations associated with those 20 amino acids. So you have the full name, the 3-letter abbreviation, the 1-letter abbreviation, depending on what we're going through. So up top, we have the primary structure, the sequence of amino acids. From there, we go to the next level and we start to form, well, I guess we'll back up there. So that top thing is the amino acid. Once that primary structure is established or created in the cell, the R groups start to fold and react to the water, whether they like it or whether they don't. That leads to secondary structure. Polypeptide chains and coils, or fold to create sections of a specific protein. So we're talking about coils and sheets. Coils tend to be hydrophobic on the inside. The R groups that are hydrophobic tend to try to group together. Sheets tend to be very hydrophilic because you're going to have water on either side of those. So going back to that diagram, you can see the sheets lining up. We got that carbon, nitrogen, carbon backbone running through each of those structures. We might have a coil or an alpha helix where the R groups that are hydrophilic will move out to interact with water. The inside will be hydrophobic, trying to get away from water. Those sheets, we're dealing with a lot of glycines. They're going to be usually hydrophilic. So different sections of the primary structure form the secondary structures. These usually happen first. Then we get to tertiary structure, where those intermediate structures interact together to form the final shape of the protein. So we have those sheets from there. So primary, secondary, tertiary, as we work our way down, those areas that are coils or alpha helixes, those areas that are sheets, are going to interact and make that protein its final shape, whatever it happens to be. So for an individual protein chain, primary, secondary, tertiary, if we're dealing with one or more chains getting together to form some sort of complex, we'll call it quaternary structure. We think about hemoglobin. It's got 4 amino acid chains interacting together. Myoglobin has 2. Muscle has thousands of chains interacting together, or quaternary structure. So different bricks making that final structure. So top of the diagram, primary, secondary structures. Those secondary structures interact together to give an individual protein chain its final shape. And then there are sometimes that these proteins will interact with other proteins to make something larger, more complex. That would be quaternary structure. So we're looking at that individual molecule there. We got primary structure on the left-hand side. We're getting that secondary structure in the middle. We've got the tertiary structure on the end. So it interacts with water. The R groups turn away or toward the water. The inside of that tertiary structure is more hydrophobic than the outside of the structure. So it's those specific reactions that are give us the protein a specific shape. So this is a protein that's probably a little bit longer in its chain. This shows you how long it takes to become folded into its final shape. So does anybody know what 2,500 nanoseconds is? 0.002 of a second. So 2.5 milliseconds. 1,000th of a second. So 2.5 thousandths of a second. We go from whenever it was made to it folding up into its final structure. So it happens very, very quickly, just like salad dressing, oil and vinegar separates out pretty quickly. It takes a while for complete separation, but it starts to happen very, very quickly. And since these things are so small, they can get that task done very quickly. So if we're thinking about this, a slight change in the amino acid sequence can affect or influence the ability of the protein to carry out its function. So we're talking about mutations, where some DNA difference will code for a different amino acid. So we may have some issues with amino acids interacting with each other. You saw on some of those diagrams, they talked about hydrogen bonding between the sheets. You have 2 chains interacting. You have hydrogen bonds holding things together. They're not fixed chemical bonds, but they tend to be a little sticky like magnets to help that final structure go together. So you've got those hydrogen bonds there. It's the reason tape is sticky. Tape is sticky because of hydrogen bonds. So those hydrogen bonds, as things stick off, different hydrogens in different locations, will help keep things relatively stable. They're not like nails, but they might be more like magnets, kind of holding things together. So shape is very important in the world of proteins. For example, genetically governed condition known as sickle cell anemia is caused by the variation of one amino acid. So you have the chain that's associated with hemoglobin. We change one amino acid in it, it changes the way it folds up and the way it interacts with the environment. So we go from normal red blood cells to sickle-shaped blood cells. They're not going to work as well from an oxygenation of the body standpoint, but they seem to confer a little bit of resistance from disease like malaria. So the character and nature of a protein is ultimately determined by the sequence of the amino acids, but really what we're talking about is the R group. What R groups in what order are going to determine how that protein comes together. So if we think about it, the R groups are part of the primary structure. The primary structure determines the secondary structure, determines the tertiary structure, determines how those molecules might interact with other protein chains to do whatever's necessary. So shape determines function. So we have that diagram there. So we're going to have 7 different classes of protein. We're going to have structural proteins, which are heavy on sheets, tend to lay out linearly, tend to be long and thin. So hair, tendons, ligaments are all structural proteins. You're going to have contractile proteins that are associated with muscle. These are very specific, very precise arrangement of proteins that allow them to move mechanically across each other. So it's kind of a ratchet as we talk about. So those contractile proteins, you have the kind of the track, and then you have the thing that's climbing up that track. Those proteins working together, you're going to find them in all 3 types of muscle. Storage proteins are more or less a place to put amino acids for a future generation. So we think about eggs, egg whites, that the chicken says, okay, here's a combination of amino acids that should be the correct building block, correct structural integrity for a chicken. So we're putting all those amino acids in that container. Hopefully we got all the right ones and there's no leftover amino acids when we're done. It's actually quite amazing that we take that little embryo in the center of an egg, and a chicken or a bird has put all of the necessary growth factors surrounding that embryo to turn that embryo into a chicken that fills, or a chick that fills that egg. So transport proteins, their job is to move things around. Hemoglobin moves oxygen around. We have transport carriers for minerals, transport carrier for fats. They tend to be, depending on their need, hydrophobic or hydrophilic on the outside because they're moving through the blood, and then whatever character they need for moving different things. So we have a charged surface that attracts oxygen and hemoglobin. In our transferon and lactoferron, we have charged on the inside that's going to attract those minerals. For our fat carriers, we're going to be hydrophilic on the outside to move through the blood, and then the center will be hydrophobic to attract those fats to want to ride inside that carrier. Defensive proteins, these are some of the biggest molecules in the body, made up of proteins, carbohydrates, moving together, but they're huge chains for defensive proteins. We're going to have some small proteins, some larger proteins associated with hormones. So oxytocin, we saw that 10 amino acid hormone that moves through the blood. And most of the body internally inside the cells is enzymes. Enzymes are the catalysts of life, the things that help get things done and directed in the right direction. So enzymes, probably one of the more important protein or roles of proteins. All chemical reactions in mammals are regulated by these enzymes. You have a shape. They talk about enzyme lock and key. The shape of the protein interacts with a carbohydrate of another protein to have something done, and then it may go back to its original shape or be permanently modified. So those enzymes are pretty important as a class, and what separates us from inorganic compounds, the ability to change, to modify, to remain not in equilibrium with the environment. So we've got nucleic acids. Is our next monomer. So we've done proteins. Any questions on proteins? Amino acids in a chain react with water to give us the shape of the protein that we're dealing with. So we'll introduce nucleic acids here. So nucleic acids, they form the polymers we know as DNA and RNA. So nucleic acids form deoxyribonucleic acid or ribonucleic acid. DNA, RNA. DNA is the code of life. RNA is what takes that code and makes it into proteins. So again, we've got all of the big four in our nucleic acids. And we got a couple other compounds as well, some sulfur, some phosphate as well, accounting for some micro or some macro minerals as part of the structure. So nucleic acids, the genetic material covering inheritance. That code is found with the nucleic acid. That's what genomics is all about. So two polymers, main interest at this point in time, DNA and RNA, deoxyribonucleic acid or ribonucleic acid. So these nucleic acids are made up of three parts, which is a little different than what we saw with our amino acids and our glucose. We have a phosphate group. We have a 5-carbon sugar. They're oxy or a deoxyribose. It's missing an oxygen. That's the one associated with DNA, or ribose, who's got that full oxygen complement. RNA and a nitrogenous base. So it looks like this. You've got the phosphate group. You've got the sugar. You've got that 5-member ring, 1 oxygen, 4 carbons. And then at a right angle to that, you have the nitrogenous base. So shape is very important. Note also the colors here. We've got yellow, blue, and then green for the nitrogenous base. That'll vary as we go through. So for the phosphate group, that is a fixed part of the chain. The sugar is fixed. That does not vary from amino or nucleic acid to nucleic acid. What varies is the nitrogenous base, and you have 5 options for that. So not as many as protein, but we can work with 5. So we look at that diagram there. We have the yellow and blue. If we think about DNA, which is what's being formed down there, we've got one side of a double helix. The yellow and blue represent the alternating phosphate sugar, phosphate sugar. DNA, that's the outside going up on both sides. You have the alternating phosphate and sugar. Then we have the different nitrogenous bases in the middle. The nucleotide is the whole thing, nucleic acid. In this case, the nitrogenous base for adenine is green. We got the other 3 there, and they always patch up A to T, G to C. And we'll start with this slide next time. So any questions about what we've done today? There you go.